Yeah, hello everyone. I will speak in my presentation about immune non-responders and do that from the perspective of an immunologist. Here first, my conflict of interest disclosures. And in my talk, I will start with the definition of immune non-responders, then speak a little bit about the proposed mechanisms of poor immune reconstitution, focusing here on the role of immune activation and also sex-specific differences in immune activation, and then end up with some potential therapeutic interventions and remaining knowledge gaps. So let's start with the definition of immune non-responders. One of the most kind of commonly used definition of immune non-responders that I will also use here is the definition of individuals who are more than 24 months on biologic suppression with bioloads under 50 copies per ml, and that then have an inadequate immune reconstitution with CD4 T cell counts under 350. So we are speaking about this group of individuals with impaired CD4 recovery under highly efficient antiretroviral therapy that leads to suppression of viremia. And there are a number of known risk factors associated with immune non-responders. One is older age, it is biological sex, it is a low CD4 count at the beginning of therapy, and it is immune activation. In my presentation, I will focus on the role of immune activation and biological sex. Let me now go into some of the potential and proposed and established mechanisms that are responsible for immune activation and some of these sex-specific differences in immune activation, and that have been linked to poor immune reconstitution. It is now established for over 20 years that immune activation in HIV infection is a bad thing. And it might not just be for immune activation, it might, for HIV infection, it might actually be in the context of many inflammatory and infectious diseases, in which we know now that elevated and persistent immune activation has negative effects on our metabolisms, on the functioning of the immune system, on cardiovascular events, and also the generation of an elevated risk for the development of cancers. These are early studies by the San Francisco group, by Steve Deeks, showing that elevated immune activation, here in that case it was measured by T cell activation, was associated with a more rapid CD4 T cell decline in the pre-antiretroviral therapy area of HIV. So these are all individuals not on antiretroviral therapy, and here in the two upper lines, these are individuals with very low levels of immune activation. And you see here that they maintain CD4 T cell counts much better than individuals with high levels of immune activation here in the third and fourth quartile that lost their CD4 T cell counts faster. Elevated immune activation is also associated with immunological non-responses in treated HIV infection. This is an important study by the Letterman lab, showing here that individuals who maintained high levels of CD4 or CD8 T cell activation, and shown in red here as failures, failed to show adequate immune reconstitution with CD4 T cell counts above 350, while individuals who successfully reconstituted their CD4 T cell numbers had significantly lower levels of immune activation, more comparable to individuals who were non-HIV infected. And the problem really is that there is a number of individuals in which elevated levels of immune activation persists despite suppressive ART. This is a larger group of individuals, showing you data here again of CD8 T cell activation in individuals without antiretroviral therapy. Here, the first three groups, you see the higher the viral load, the higher the level of immune activation. So immune activation is already lower in individuals that have low levels of viral load without antiretroviral therapy. Antiretroviral therapy that suppresses viral replication further decreases that level of immune activation, but it remains higher than individuals who are HIV negative And it is in particular this group here of individuals with persistently elevated numbers of T-cell activation despite viral suppression that have an increased risk to become immunological non-responders. The problem really is that there are limited options for these individuals with immunological non-responders. We know that these persistently reduced CD4 T-cell counts and immune activation are both associated with increased risks for AIDS and non-AIDS related morbidity and mortality. So we want to do something. However, several studies have shown that adding antiretroviral drug to already suppressive antiretroviral therapies or ART intensification is not really improving CD4 cell recovery or reduce immune activation in most studies so far. So there are no clear recommendation to do so. And these are here the most recent guidelines from 2019, who were updated in 2021. Switching antiretroviral drug classes is also not shown to consistently improve CD4 T cell recovery or reduce immune activation, and is therefore not recommended. There were larger IL-2 trials that had shown that they can increase CD4 T cell counts, but however, there didn't show any clinical benefit in terms of reducing AIDS-related and non-AIDS-related morbidity and mortality. So this is not recommended as there is no clinical benefit. And also, there's no other clear interventions that have been shown to decrease immune activation in this group of individuals. There are a number of explorative approaches, blocking inflammatory cytokines such as IL-1 beta, for example. However, there have been no clear clinical hard endpoints on this yet, and there's no clinical approach approved and proven to decrease morbidity and mortality during in these individuals despite that have antiretroviral mediated viral suppression. So that one is left pretty much with general efforts to reduce these comorbidities, such as encouraging smoking cessation, healthy diet, regular exercise, treating hypertension, hyperlipidemia. So things that we would actually recommend anyone for a healthier lifestyle. And these limited options show us that there is clearly the need to develop novel treatment strategies. And for that, we need to understand in more detail the underlying mechanisms that are responsible for this elevated immune activation. So what can cause immune activation in HIV infection? There are a number of factors. It can be the virus itself. There are co-infections such as CMV that have been described. There's the impairment of the gut barrier that can lead to microbial translocation that has been linked to immune activation. And there's also the thrombosis of the lymph nodes and other secondary lymphoid organs that has been associated with immune activation. In general, in an HIV infection, there is a depletion of the CD4 T cells in the mucosal layer that can lead then to impaired integrity, microbial translocation, including LPS, that can signal for TLR7, induce the secretion of pro-inflammatory cytokine, leading to immune activation. Furthermore, HIV itself, in particular, the single-stranded RNA within the virus, can be sensed by TLR7, another toll-like receptor, that then can also induce the production of pro-inflammatory cytokines and type 1 interferons that lead to immune activation. So how is HIV recognized by the innate immune system? Apart from these toll-like receptor pathways, there is an increasing knowledge we have gained over the last years, and this is kind of constantly added to this. We have a number of innate pathways that can sense the virus, either capsid or other components of the virus, and then trigger the production of pro-inflammatory cytokines, the production of type 1 interferons that further trigger the induction of interferon-stimulated genes. And ultimately leading to immune cell activation. There are also other pathways listed here that can activate the caspase pathway, leading to pyroptosis and cell death that can be a major inflammatory signal. Also, the induction of IL-1 beta contributes to these inflammatory responses. And here again, the toll-like receptors play an important role. Toll-like receptors such as TLR4 and TLR7 are located on many of our immune cells. Toll-like receptors that sense bacterial components such as LPS on the outer membrane, toll-like receptors that sense oligonucleotides such as single-strand RNA here in the endosomes, and they then trigger a very complex signaling pathway that leads to the induction of these inflammatory cytokines. And as we understand more about the signaling molecules that are involved, there's actually an increasing drive to develop drugs that can focus on the suppression of these pathways, more specifically, as these pathways also play a role in many of the inflammatory diseases and autoimmune diseases. So I will focus here in the rest of my talk on TLR7 and 8, who can sense single-stranded RNA and have been shown to be able to sense HIV. Does HIV encode for these TLR7 and 8 ligands? It does. This is just showing you the single-stranded RNA of HIV. There is this one TLR ligand that was early described here, that because of this high uridine content can trigger TLR7 signaling. And there are just many of these areas within the single-stranded HIV RNA that can serve as TLR ligands. And several years ago, we tested them, we synthesized these uridine-rich areas of HIV and stimulated PBMCs with that, and observed a significant induction of interferon alpha, in particular by PDCs. And many of these uridine-rich areas within the single-stranded RNA can actually induce potent interferon alpha production by PDCs that express many of these strongly expressed TLR7. And this production of interferon alpha and other pro-inflammatory cytokines that is triggered by TLR can then induce a secondary activation of T cells and NK cells. So what we know now that HIV can be sensed by several innate immune pathways, including the TLR7 pathways, induce the induction of pro-inflammatory cytokines such as type 1 interferon that then triggers other parts of the immune system, including T cells, and induces the expression of these activation markers on T cells. Interestingly, the ability of the female and the male immune system to respond to TLR7 and TLR7 stimulation differs. PDCs from women produce significantly more type 1 interferon in response to TLR7 stimulation than cells or PDCs derived from males. And this is quite specific for TLR7, as we do not observe these differences for TLR9. And we also looked at this in the context of HIV-derived TLR7 and 8 ligands and observed that PDCs derived from females produce significantly higher levels of type 1 interferon than PDCs for men. What are the mechanisms for that? The TLR7 gene, or the gene encoding for TLR7, lies on the X chromosome. As you know, men have one X chromosome, while women have two X chromosomes. And while for a long-term time we thought that the second X chromosome is inactivated, newer studies now show that actually the TLR7 gene can be read from both X chromosomes, and there's actually escape from X chromosomal inactivation for TLR7. You can track that when you use an individual who has a SNP here on the second X chromosome, so that you can differentiate between the gene or the RNA transcribed from the one X chromosome and the other. And in a man who only has one X chromosome, obviously all the RNA comes here from one of the alleles of the X chromosomes, while in women, we can consistently observe a group here shown in green of cells with PDCs that actually contain RNA that is coming from both X chromosomes. Here on the single cell level, we have escape from X chromosomal inactivation. And the cells that transcribe TLR7 from both X chromosomes have also significantly higher levels of mRNA expression and protein expression. We also showed now that if we then trigger them with TLR7 ligands, these cells that actually transcribe TLR7 from both X chromosomes, we have significantly higher production of type 1 interferon. So in general, TLR7-mediated sensing of HIV can induce immune activation, and that differs between the sexes, where in women we have much higher production of type 1 interferon, higher induction of interferon-stimulated genes, and in males we have less of that, and that might explain some of the sex differences described between men and women in acute and chronic infection. We know now for many years that women with the same viral load as men have a higher risk to progress to AIDS and to lose their CD4 counts. And this might be due to the fact that there's a lower threshold for sensing HIV RNA that then triggers higher levels of immune activation in women than in men, and this higher immune activation leads to faster loss of CD4 T cells in untreated infection. We also know that women in primary infection actually control primary HIV replication better. This is data from a large cohort of over 2,000 individuals, where at any age in this primary infection setting, women had lower viral set points than men. There's about a half log difference here. These differences in immune activation might actually, between females and males, might actually also already play a role in utero HIV transmission. We recently, in collaboration with our African colleagues and Philip Goulder's group, observed that but women who get HIV infected during pregnancy with primary infection and therefore have a higher risk of transmitting the virus to their infants do so significantly more frequently to female babies in utero than to male babies in utero. We were able to show that there's also in these female newborns already a higher level of immune activation in response to the maternal infection that might actually then increase their risk by more activated CD4 T cells to become infected. And in collaboration with Eileen Scully in Steve Deek's lab, Eileen recently described that also in individuals who are on suppressive heart, there are sex differences, and that there are lower levels of residual immune activation in women on suppressive heart, and that was also linked to lower reservoir sizes. So in general, we know that sex matters in HIV infection, and that in females we observe better control of varying in acute infection, however, faster progression in chronic untreated infection that is due to higher immune activation. We also have differences in immune activation between females and males in treated infection, and it might also have a risk on a higher risk for in utero HIV acquisition. So with this knowledge, what are potential future interventions and remaining knowledge gaps? I think in general, we have made progress to understand how a virus is sensed by the innate immune system, what pathways are triggered, and how that induces immune activation. However, there are still many gaps here in these pathways that we don't fully understand. But what it is of interest is that there are now an increasing number of drugs that can precisely target these different, several of these molecules involved in the signaling, cytokines, inflammatory cytokines that are secreted. And it's important now to use this knowledge and do clinical trials to understand whether blocking some of these pathways involved in HIV-induced immune activation can reduce immune activation, help CD4 reconstitution, but then also actually have clinical benefit, as we saw that the IL-2 treatment trials can enhance CD4 T cell counts, but did that without a clinical benefit. So in conclusion, elevated immune activation is associated with immune non-responses in individual on suppressive heart. Immune activation and immune non-responsiveness, or no good reconstitution of the CD4 T cells, are associated with an increased risk of AIDS and non-AIDS associated morbidity and mortality in these individuals. there are currently limited treatment options for individuals with compromised immune reconstitution. We now understand the immune pathways, in particular the immune pathways that play a central role in HIV-associated immune activation. And these can also account for some of the sex differences we see in immune activation in HIV reservoirs, but also in the risk for INR, and a better understanding of the molecular pathways involved in HIV-mediated immune activation of the innate immune system forms the basis for the development and testing of novel therapeutic interventions aimed at reducing immune activation and INR. So I end here. I want to thank members of my team here and individuals who contributed some of the data that I presented in my presentation, and the funding agencies that fund our research. And I end here, and I'm happy to take questions.